Hello, and welcome to another lecture of Psychology 101. I'm your instructor, Dr. Shadi Bashai, and in this lecture, we will continue talking about psychological approaches to research methodology, and that corresponds to Chapter 2 of your textbook. In the last lecture, we ended our discussion speaking about internal validity and external validity. Internal validity is defined as how much confidence I have that the change that I observed in my dependent variable could be attributed to the manipulation I did and or the change that I did as a researcher in the independent variable. The less confidence we have that the change in the dependent variable was due to change in the independent variable in the experiment, then the lower my internal validity. There are several systematic ways which internal validity can be threatened. Okay? And we will talk about a few of these systematic threats to internal validity right now. And the first of which is this idea of maturity. Okay? And for the next, in speaking about the threats to internal validity, what I'll do is I'll set up a particular example here for you, just to make things concrete. Imagine we are examining the effects of an educational program on educational attainment for children with learning disabilities. And how we study this is we get a bunch of students who have issues related to a learning disability or a learning dysfunction. And then we look at their educational attainment or grades, let's say, at baseline. And then we examine, and then we administer the intervention. And then we examine their educational attainment again after the conclusion of our intervention, and to see if there have been any gains that have been experienced by these children. Okay? Well, let's say that I do find an increase in grades from time 1, which is baseline, to time 2, which is post-intervention. That could be, of course, due to my intervention, but it could also be due to a number of other factors. One factor that kind of makes us have less confidence that the intervention changed the outcome variable or the dependent variable, which is educational attainment or grades, is this idea of maturity, meaning that from time 1 to time 2, my participants have grown. They've developed in some way, okay, that have very little to do with the intervention in and of itself. It could be just natural development, natural growth that happens over a period of time. And that could actually be why the students are actually experiencing the educational gains that they did experience throughout the duration of the study, as opposed to the intervention in and of itself. There's an idea called the statistical regression, which is another systematic threat to internal validity. Okay? And the idea of statistical regression towards the mean is this idea that if you get a bunch of participants who are showing higher or lower averages on a particular trait, and remember, in our example of children with learning disabilities, these children would probably be getting lower grades than most other people in that institution or that university or that school. But what happens over time, through this concept called statistical regression towards the mean, is that people naturally slide from an extreme position to more of what the mean looks like for the population. that is extreme positions, extreme scores, don't stay extreme for long. They regress back towards whatever the population mean actually is, just by sheer passage of time. Okay, so even though we are seeing a shift in educational attainment over the period of the intervention, it may have very little to do with the intervention itself, but just simply people sliding back towards whatever the mean is for the population. Another threat to internal validity is this concept of selection. Let's say, for example, that I allow people in my study. I only let people in who've signed up within 2 or 3 weeks of me posting about the study. Okay, and I find that over the course of the intervention, over the course of the study, the students that I allow, the participants that I allow into my study have actually seen tremendous gains over the duration of our assessment. And I could wrongly conclude that the change observed in their educational attainment was actually due to the intervention, when in fact it could be something about that group of people in and of themselves that's making them interact better with this particular intervention. Okay, that is, there could be some kind of a selection bias here. It could be that the people that arrived first have something special about themselves that would allow them to kind of feel this nourishment during the duration of the study. So again, that's a threat to internal validity, because we look at the scores from time 1 to time 2, and we see a change, a positive, almost a hypothesized change in the right direction, but we wrongly conclude it's because of what we did, as opposed to some other unknown variable that we simply didn't control for. Another concept is attrition. If you're looking at people's scores over time, what ends up happening is people drop out of the study for whatever reason. People move. Unfortunately, sometimes in health-related research, people pass away. Okay? And if that happens, if people drop out systematically out of your study, there could be a reason why they're doing that. For example, extremely severe cases could be prematurely dropping out of your intervention, and at the end of the study, you're seeing that people that weren't so severe actually performed fairly well, and then you conclude that the intervention works for the population of students with learning disabilities. Well, that's not actually true, because you had other extreme cases in your study that simply dropped out, probably because they're too severe of a case. And for that reason, your results at the end are relatively skewed. They're only based on a subset of your original sample that you began with. Okay, so these are all threats to internal validity. So we talked a little about what an independent variable is. We talked about what a dependent variable is, which is the thing that we observe, and we think something happens to it based on changes we make in the independent variable. But there are also other basics of experimentation, including participants. And of course, I've been using the word participants throughout the lecture so far. And participants are basically the individuals who take part in our experiment. And these are the same individuals whose behavior we're really interested in. So these are the individuals whose behavior we observe, whose behaviors we monitor, whose behaviors we record over time to see if in fact our manipulation had any effect at all. There are other concepts. Okay, and we talk a lot about experimental group, and we talk a lot about control group, and these are of course two different or separate concepts. In most trials that intervention-based trials, for example, there's usually a group that receives the manipulation And then there's another group to which we compare the results of the manipulation against. And let's go back to our example of an intervention for students with learning disability. Let's say instead of putting the entire sample, my entire sample of participants, through the intervention arm of my study, what I end up doing is that I split them equally into two groups. Let's say I randomize them into two groups. And the one group I give the intervention to, and I observe and I record their performance from time 1 to time 2. And another group that I simply don't do anything with, and I keep them simply on a wait list, and I also look at their academic achievement from time 1 to time 2. Okay? So in this particular example, the group that receives the intervention is the experimental group, whereas the other group, the group that we're just recording, just to compare the results of the experimental group, is called the control group. Okay? And usually, the control group does not receive any intervention at all, although stronger designs, what they'll do now is they'll probably give a placebo, some kind of a placebo pill. Let's say if it's a drug intervention that we're talking about, and if it's a psychological intervention, there are other sham types of interventions that don't have any active ingredients in them that we can use as control, again, that is used as a comparison group by which we compare the results coming from the experimental group. There are other basics of experimentation that we haven't yet talked about. And one of the one that is a very important concept in the area of experimentation and research methodology is this concept of sample size. Okay? And there's a rule of thumb in experimentation which says that the higher your sample size, the higher the chance that you will actually find whatever it is that you're looking for. That is, if your sample size is sufficient, it's a big enough sample size, then the study or the experiment is said to be powered in order for you to detect a difference between the groups if a difference actually exists between them. Okay? The lower the sample size, the lower this concept called power. Sample size has a very interesting and unique and intricate relationship with something called effect size. And effect size basically says that not only is there a difference between group A and group B, but the difference is massive, let's say. Okay? So it's not a binary, yes, there is a difference, no, there is no difference. It actually gives you the degree of how much difference there is. The higher the effect size, that is the bigger the difference between the groups, the smaller the sample that you can get away with. Okay? Because you simply just don't need as many observations to see that effect. So we talked about power. Okay? Power is the ability to detect whether or not there is a difference between the groups if one actually exists. And for a study to be amply powered, you need a sufficient enough sample size. And statistical significance talks about whether there is a difference or not. Okay? And then there's a concept called effect size, which is the degree of a difference if there is actually one. And then finally, there's a concept called clinical significance, meaning not only is there a difference between group A and group B, and not only is the difference large, it actually matters in the real world. It has a real world application. It'll solve a problem that we're currently facing. So we've been talking about all the steps of the research method. And now we've arrived at step 4. And in step 4, once we've picked our design, we have our question in mind, once we've decided on how big the sample is, how we're going to measure whatever it is that we're interested in measuring, what the dependent, independent variables are, how many groups there are, whether there is an experimental or control condition, right? So we picked the design, did all of it. Now, I have to decide on what techniques must I use to actually analyze the data that I have. And again, this is firmly within the quantitative research bracket or world, okay? And if we're doing quantitative research, we have to find a way to work with our data, work with our numbers, to answer some of these important questions that I started out with. And some of these important questions are, is there a difference, for example, between the experimental group and the control group? And let's say this is a trial looking at a brand new drug for depression. Is there a difference between the experimental group, meaning the group that gets the new drug for depression, and the control group, the group that is receiving a placebo pill, an inert pill that has no active ingredients? Is there a difference between them on depressive symptoms from time 1 to time 2? Okay, and in order for me to answer that question, I have to subjugate my data, my numbers, the numbers that I gather throughout the process to the statistical process in order to answer this question. But it has to fit with the right type of data, and it has to fit with the right type of question that I'm interested in asking. And there are many, many statistical techniques that we can discuss together. And then finally, once you have your results, you've ran the appropriate statistical analyses, etc., now you're ready to actually figure out whether your hypotheses have been supported or whether your hypotheses have been refuted or disproven. Okay? And once I figure that out, I put all of it, including the question, including the background behind the question, why I was asking that question to begin with, why is this a problem even to begin with. I put all of it in a research report that follows a very kind of a strict structure, if you will. Okay, and that strict structure usually is you start out with an introduction, and then your methodology. Tell us how exactly you're measuring the things that you're measuring. What exactly are you measuring? Who are you going to recruit for your participants, so on and so forth? Where are you recruiting them from? And then your results, which is exactly the step that we talked about just before this one. And then finally, your discussion section, in which you talk about whether your experiment worked, you found a difference as predicted, or not. Maybe you didn't find an effect that you were looking for, but why? How do you explain this through all the research that's been done on this particular topic? Okay, and once you have this report prepared, then you can actually try and publish it. And the way it works is you submit your report to a journal, which usually has an editor, and then the editor takes a look at the subject matter. Let's say it's on a study on depression, and then the editor takes your report and sends it to people that he or she deems as an expert in the area of depression. And the reason for that is, and if you remember, one of the cool things about science, and it's something that differentiates it from pseudoscience, is this idea that all of our ideas, all of our findings, all of our experiments, before being released to the scientific public, to the scientific community and to the public, all of them have to be vetted through a number of experts in the field. And this is how we do it. So the editor will send it out to usually 2 or 3 expert reviewers who will then read the document very clearly and very carefully and they will send in their review to the editor, who then makes a decision whether or not this is good enough as is to make it into the journal, as a published product. So we talked a little about all the kinds of different statistics that can be used. And remember that we need to, if you remember this step, we need to subject whatever our data is, whatever the thing that we collected, basically, to a particular test that matched the question that I began with. But there are basics of statistics that are important for us to discuss right here. Okay, so statistics, of course, is a branch of mathematics that is very much interested in classifying and analyzing data. It is by far the branch of science most used for the behavioral sciences, such as psychology, sociology, etc. There are two branches of statistics. One that's called descriptive statistics, which is simply a way to summarize the findings. It's a very, very quick and dirty way to tell you what exactly is going on with your data. And then there's another branch called inferential statistics, which is a bit more advanced forms of statistics. And the idea behind inferential statistics is that you're actually trying to answer a question related to, is there a statistically significant difference between the experimental group and the control group? Okay, so that's the branch of inferential statistics. It tells you and it gives you an inference about a particular population. Okay, so is let's say I'm doing an intervention with anorexia, let's say, and I have a group of patients who suffer from anorexia, and they're randomly assigned into the treatment or the experimental group or the control group. And then I run my statistics, etc., after I give them the intervention, and then I find actually that the experimental group, the group that received the intervention, is showing much lower anorexia-related symptoms by the end of the intervention. Inferential statistics helps me figure that out and tells me that, yes, the population of people with anorexia would likely respond to this particular intervention if all the other steps have actually been honored. Which is, what I mean by that is, if there is true random selection, if there's true random assignments between conditions, etc., inferential statistics is going to give me an answer as to whether the experimental group, which theoretically should look very similar to the population of, the big population of everyone with anorexia or anorexia-related issues, whether that experimental group will respond to the intervention, and understanding that will give us an indication whether the population generally, the population of people with anorexia, would respond to said intervention. So we talked about descriptive statistics, which is a simple way of summarizing or describing the data that I have. And there are three very important types or concepts within descriptive statistics, and we call these concepts central measures of central tendency. Okay, and the first is the mean, which is basically the average of all the sets of scores. So I'll give you an example of what that might look like. Let's say you have three observations. Okay, a score of 3, and a score of 5, and a score of 8. Altogether, of course, drum roll, these are 16, okay, which is 8 and 8, really, if you think about it. So 16, and then you divide by the number of observations there are. So it'd be 16 divided by 3, whatever that number might be 5.3 is really what the mean of that particular data set would be. And then there's the mode, which is the most frequently occurring score in a data set. let's go back to that example that I gave. Let's say there's a 3, there's a 5, there's an 8, and then there's an 8. There are 4 observations now instead of 3. In this particular 4 observation data set, the mode is going to be 8 because it is the most frequently occurring number because you have 3, 5, 8, 8. The median, on the other hand, is the number that exactly splits the data set down the middle. So exactly 50% of the scores fall below and 50% of the scores fall above that particular median. I'll give you that same example. You have 3, 5, 8, 8. Let's add another one there. So 3, 5, 8, 8, 10. So these are 5 observations, 3, 5, 8, 8, 10. The median here would actually be the first 8 because it is the number right smack in the middle of my data set. There are 2 observations before. You've got 3 and 5, and then there are 2 observations above, 8 and 10. This is the picture on the left-hand side in the corner there. This is what we call a normal distribution or a bell-shaped distribution. Most human traits, including height, including intelligence, including impulsivity, including conscientiousness, whatever it might be. If you go out and examine these particular traits at the population level, you will find that scores will be distributed in this normal bell-shaped distribution. That is, you have exactly 64% of the sample, so 64% of people will fall somewhere around the average of whatever that trait is. So the majority of the population is going to be clustered around the average, and in the photo that I have up here, the average here is 0. And as you'll see, there is something we call a standard deviation, and these are the numbers along the bottom here, which is the 0, 1, 2, 3, and or -1, -2, -3, of course, the other way, the left side. And standard deviations are the average spread away from the average. The majority of people's responses on most human traits will fall within one standard deviation of the mean here being 0. So they'll fall anywhere from -1 to +1 in that distribution, and it is by far the majority of people, be 65% or so. And then the more extreme the trait, the less and less people you'll find who have those extreme levels of whatever trait you're interested in, including intelligence, etc. Okay. A positive skew, which is the graph on the right-hand side, basically tells you that the majority of the data is not so much happening in the middle of the distribution, which was the case in a normal distribution, but a positive skew actually signals that there's actually more data closer to the left-hand side of the graph. Okay, and if you want to remember positive skew, positive is almost always there's more data on the left-hand side, and then a negative skew or a negative skew to the data will be there's simply more data on the right-hand side of the graph, and you see a depiction of that in graph B, which is at the bottom of your screen there. And the concept here is there are more observations that are happening near the tail end of whatever distribution, either be positive or negative. So C or B. The majority of observations are not happening in the middle. There's way too many people with smaller levels of that trait, or way too many people with large levels, high levels of that particular trait. Whatever trait you're thinking of. And it doesn't happen often that naturally occurring traits, human traits, intelligence, IQ, whatever it is, IQ, impulsivity, etc. It doesn't really happen often that they're either positively or negatively skewed. Most often you'll see them in a normal distribution, as you'll see, as you're seeing in the graph on your left. If you want to challenge yourself a little bit, I recommend you stop the video currently and calculate the mean, median, and mode for the data set that I have up here, which is 88, 85, 86, 81, 87, 80, and 87. Okay, and the first thing I would recommend for calculating median specifically is to actually put these data sets or these observations in order, in the order that you're seeing them, from lowest to highest. So we talked about independent variables, we talked about dependent variables, we talked about threats to internal validity. We haven't really talked about placebo effect. I mean, I've mentioned it in a couple of examples, and the placebo effect is something that pretty much all psychologists working with interventions, so working to develop particular interventions for particular disorders, for example, as well as health researchers, if they're developing interventions for particular health disorders, cardiovascular disease, cancers, whatever it is. It's all of these people have to contend with this concept called the placebo effect. Okay? And the placebo effect is simply the expectancy that something will actually happen in the experiment. So it's the participant's expectation to see a positive change in the dependent variable based on their belief that they're taking part in a study. Kind of similar to the Hawthorne effect, but a little bit different. Okay? So it simply says that when people expect to get better, they will. And for that reason, participants themselves, let's say we're designing a new drug treatment for anxiety, let's say social anxiety disorder. Okay? And we have two groups of participants that I've randomly assigned into these two groups. In one condition, in one group, they receive the actual new drug that I've just developed. And in the other condition, they receive a placebo pill. And a placebo pill is a sugar pill. It is an inert pill that has no active medicinal ingredients. And the reason why we do that, and the reason why this is important to do, especially in all kinds of health or clinical psychology research, is, like I said, people's expectations can in fact produce change that has very little to do with the intervention itself or the active ingredients in the intervention. For that reason, we typically don't tell people which group that they're in. That is, we give them a sugar pill without actually knowing that they're taking a sugar pill. And when that happens, we say that participants are blinded to study hypotheses. They don't know what exactly we're expecting. They don't know which group that they're in. They know that this is a study on a new intervention for social anxiety, and they genuinely believe that they could be an experimental or the control condition. They have no way of knowing. So that's placebo effect, is the expectancy that things will get better. And then there's the nocebo effect. This is actually the opposite of the placebo effect, which is the belief that things will just not work out. Okay? And again, both the placebo effect and the nocebo effect are powerful concepts that can actually skew people's data and makes us less confident that whatever we observed was actually due to whatever intervention that I administered to the group. There's the experimenter expectancy effects, again, which is another important pitfall of experimentation. And the experimenter expectancy effect is when, let's say, I'm the person that designed this new intervention for social anxiety, this new drug treatment for social anxiety. And let's actually imagine for a second that I'm the one doing the monitoring of symptoms of all the participants, either they be in the placebo condition or the actual drug condition, the actual intervention condition, the experimental group. So let's say I do these assessments, but I know which group is which. I know which participants belong to which group. But there is something subtle that I do when I make these observations. Let's say for all the participants that I'm assessing who are actually in the experimental group, the group that is actually receiving the treatment, I start acting very kind and not pushy at all. And again, I don't do this knowing that I'm doing this. It's just very subtle way of, or at least very subtle behavioral changes that I do to make people in the experimental group feel a little bit more comfortable. Again, this is inadvertent. That is, it's beyond my awareness that I'm doing this. But it can and it does sometimes skew the data in a very important way. That is, you're behaving towards the experimental group in a way that fits your expectations. Okay? Let's say when you're recording down their symptoms, you're not really listening to what they're saying, so you listen to their symptoms through a rosy-colored glasses, because again, this is what you want to be true, is for the drug to work. So you start ignoring all the signs that maybe, just maybe, the drug isn't working for these people. Okay? And for that reason, sometimes the people who put forward the hypotheses, that is the experimenters themselves, the researchers themselves, they hire an army, a team of people to create this barrier between the researcher and between participants in the study. And this is precisely for the reason that you don't want to start behaving as a researcher in ways that would confirm your hypotheses to begin with. You want to be able to say that whatever difference I observed in my study, it wasn't for the reasons of me changing my behavior around participants. And then finally, there are demand characteristics, which is another pitfall of experimentation. And demand characteristics are any obvious signs of what the hypotheses are and or what the researchers are expecting. Okay? So any outward sign to participants related to what the hypothesis might be or what the researcher is expecting to find might actually make participants in the study behave and or disclose of symptoms or behaviors that fit with the expectations of the research. That is, people will subtly start changing their behaviors or the things that they're actually telling you to fit whatever expectation the research has for them, if there is enough information there. For that reason, sometimes we employ what is called a disguise. And these disguises basically, they're a way to mask the true nature of the experiment, in order to really get an accurate and unfiltered view of the behaviors and or the emotions of the participants in the study. That is, if you give away too much, if there are way too many demand characteristics, there could be a problem because people start behaving in ways to confirm whatever the demand characteristics may be. Okay? And of course, using a disguise, using some kind of a lie to mask what the true nature of the study is quite controversial, and it needs to be wholly justified before researchers do that. At the end of this lecture, we will talk about a very dark history of science, if you will. And that dark history centers around studies such as the Tuskegee syphilis study. And the reason why we bring this concept up is we as researchers have throughout history abused the powers that we have. We've used the data inappropriately. We've done things that are inhumane. We've done things that are simply wrong and unethical. And for that reason, there are ethical guidelines put in place nowadays, and they're constantly being changed and constantly being updated, that make sure the research is being conducted in an ethical and in a moral manner so that we're actually not harming our participants. One of the very first steps in the process of ethical research is obtaining what is called informed consent. And this is when we provide participants with enough information about what the study will entail to get them to make an informed decision as to whether or not they want to be part of this research as participants. Okay? So it's really giving them enough information to be able to make their minds freely as to whether they want to be a participant in our research. And there is a bit of tension here between a concept called informed consent and the concept called demand characteristics, which we talked about a little bit before. So informed consent, as I mentioned, is giving people enough information about what the study will entail so that they can freely participate. And demand characteristics is when you give too much away as to influence the participants' behavior in a way that the research isn't designed to catch. That is, if you give them a lot of information about what the hypotheses of the research might be, people may inadvertently, as people do, change their behavior or change their responses to fit with these hypotheses. And again, this all happens inadvertently, without people's knowledge and beneath the threshold of awareness. So the first step of the process of ethical research is informed consent. The last, or at least one of the last steps in the process of ethical research is the concept of debriefing, which is basically happens at the end of the study, and this is where you spill all the beans about what exactly the study was about. So this is where you give people, and you become totally transparent about what your hypotheses were, about what your independent variable was, about what your dependent variable was, so on and so forth. And this really provides the participant with enough information, but also a means to ask questions and to gather a bit more information about the type of research that you're doing. So ethical guidelines, as I mentioned earlier, are always being updated, but they apply to all clinical psychologists, and they apply to, obviously, their clients and to experimental psychologists and their research participants. And big governing bodies such as American Psychological Association, or APA, and Canadian psychology governing body such as the Canadian Psychological Association, as well as the funding agencies within Canada that are national-based funding, such as SSHRC, NSERC, or CIHR. All of these have their own ethical guidelines as to how to conduct appropriate and ethical research with participants, and all psychologists must abide by such rules. So there are a number of controversies in ethics. One big controversy, and this is done to to mask demand characteristics. And remember, demand characteristics is when you give too much away about what your research is actually about, as to actually influence the participant's behavior in a way that you don't expect and you don't want. So lots of researchers now, and have been for a very long time, they've been using something called deception, meaning there's some kind of a lie that masks the purpose of the research. And I think I was alluding to that a little bit earlier. Okay? So some people have wondered whether this is appropriate. And on one side, the proponents of using deception, they say, well, the use of deception is actually necessary in order to get at certain questions that if you give a lot of information to people about that certain question, what they will do, as I was mentioning earlier, it'll be too much demand characteristics, and they'll start changing their behaviors in a way to render your ability to solve the question impossible. Okay, so for that reason, they inculcate, if you will, the research question in a ruse, in a lie, in a deception. So that's the proponents of that approach. The opponents of that approach say, well, we understand why there is a need for to use deception, but the use of deception is more harmful than it is good, in that it may actually harm people's views of psychology and psychologists. It may inadvertently make people feel awkward or have some negative emotions about the fact that they were just lied to in the research. So that's one big controversy, whether or not to use deception. Another big controversy is whether or not to use animals in research. And about 7 to 8% of all research in psychology uses animals. And proponents of this say that much like research with humans, we have ethical guidelines to guide appropriate research with animals, and the majority of animals who are used in psychology research are rodents and birds. And they tell you that that's the only way, really, to get at a number of fundamental questions that we couldn't otherwise answer if we're strictly using humans, because it would be the consequences would be far too dire. And I'll give you an example of what that looks like. So a lot of research, for example, is being conducted on brain plasticity and recovery after brain injury, and that is mostly done with rodents. So the use of animal models is really prominent in this field. Okay? And some people would argue that to create a controlled experiment with rodents and measuring how brain injury may recover over time is the only way to do it, obviously, given the fact that we can't do that in a controlled setting with human beings. It would be far too unethical. So that's the proponents. The opponents say animal models may not translate very well to humanity. So they question the validity even of that research coming out of animal laboratories. And B, is that we're abusing our power over these animals. Okay. So why am I droning on here about ethics in psychology research, ethics in research generally? And that's because we've abused our powers as researchers. And this has happened time and time again. And there's probably nothing more illustrative of this abuse of power in research than the Tuskegee study, which was a study that ran from the 1930s to the 1970s. And the study was done with mostly low socioeconomic status black men who suffer from syphilis. And the point of the study was to examine the effects of syphilis on the brain. Well, that's good and dandy until in the 1940s, syphilis was actually cured. So penicillin was discovered and syphilis was cured, but the problem was the researchers in this particular study, the Tuskegee studies, they did not provide the patients in their study with penicillin even after its discovery, just to see how far or what the extent of the damage syphilis would do on their brains. Absolutely abhorrent stuff. So this is why we need ethical guidelines to guide our research so that we don't abuse our powers. Until next time.